Welcome back to a new video session from Biology Boost Up. Today in this video tutorial session we will study about the mechanism of DNA replication. In my previous video lecture I already had talking about the basic concepts regarding the replication. So this is my request to all of you, please visit my previous lecture. I will give the link of video in my description box. So now let's start. At first, the initiation of replication at the specific region of DNA which is known as origin of replication or ORIC. Here the DNA A protein comes and helps to unwinding of the DNA. Then helicase or DNA B and DNA C protein comes and combined with and separated the two strands of DNA. Now those separated two strands act as a template strand and form a balloon shaped structure which is known as replication bubble. The DNA A, DNA B and DNA C protein forms the pre-priming protein complex. Now with the pre-priming protein complex, DNA guidase or topoisomerase 2 and SSBP makes a complex that is known as the initiation complex. Here the topoisomerase helps to opening of supercoil of DNA helix and SSBP covered the both separated strand to prevent rewinding of each strand during replication process. After the formation of replication fork by the help of DNA primase, a small fragments of RNA which is 5 to 15 base pair long is synthesized over the template strand of DNA. These small RNA contain a free OH or hydroxyl group at their 3'. Next, the elongation process. Now, after the formation of replication fork, the DNA polymerase 3 helps to attached of deoxyribonucleotide monophosphate with the free OH unit or group at the 3' end of RNA primer. This deoxyribonucleotide monophosphate always being complementary with the template strand. The DNA polymerase 3 attached those complementary nucleotides one after another. As a result, the strand become elongated. The DNA polymerase 3 synthesized new DNA from the 5' to 3' direction. Hence, the 3' to 5' template strand is polymerized by the DNA polymerase continuously in 5' to 3' direction, which is known as leading strand. Now, the another strand of 5' to 3' template is polymerized in a frequently interrupted manner and produce a short fragments of DNA that is known as Okazaki fragments as helix unwind in 3' to 5' direction of the template. Thus, this strand is replicated discontinuously in 5' to 3' direction called lagging strand. The primosome helps to production of these Okazaki fragments. Now, when the replication moves forward, DNA polymerase 1 removes the RNA primers from the template strand, which are attached with the Okazaki fragments, and DNA polymerase synthesized complementary nucleotides over the lagging strand of DNA. The ligase enzyme joined those fragments of the DNA by the formation of phosphodiester bond. Now, the termination of DNA replication. When each replication fork of replicon fuse with each other, then the replication process becomes terminated. After the termination, two copies of DNA are formed. Each parental DNA forms the hydrogen bond with the newly synthesized DNA strand. This is the diagrammatic overview of DNA replication, and you can see here all those different enzymes which are involved during this mechanism. So, to better understand, I request to all of you, please view my previous lecture regarding the protein and enzymes which are involved in DNA replication, and the link of this video I will give my description box. Thank you. Please like, share and subscribe the channel.